Very good morning, students. So in this video, you're going to learn about organ culture, how it can be done in plant tissue culture. So let me share the PPT to start the content. Okay. So what do you mean by organ culture? Any plant organ can serve as an explant to initiate your plant tissue culture. We call it as organ culture. So here are few examples. If you have taken shoot as your explant, that is also one plant organ. We call the plant tissue culture as shoot tip culture. If root as an explant, if you have taken that culture, we call it as root culture. Leaf culture. Flower, since flower is a reproductive organ, you can take both male reproductive organ, anther or pollen, female reproductive organ, ovary, ovule also. So whenever you're using a complete plant organ to serve as an explant to initiate the plant tissue culture, that we call it as organ culture. So moving on to the technique, haploid production. Haploid production is nothing but when you are producing plantlet by plant tissue culture characterized by possessing only a single set of chromosome, that we call it as haploid production. Say, for example, if I'm using anther, immature anther to initiate my plant tissue culture, the plantlet which I get only having half set of chromosome. That we call it as haploid production. So this technique, why we are doing is it having a great significance for the production of homozygous line. Homozygous line is nothing but having only single set of chromosome. Later, based on my wish, I can cross this chromosome number, half chromosome number with any polyploidy, aneuploidy to increase the plant genetic engineering technology or plant breeding technology. That's why it is in use. So this technique can be done two ways. Androgenesis and gynogenesis. Androgenesis is nothing but if I'm using the male reproductive organ, anther or pollen, andrecium, if I'm using to raise my haploid plant, means that culture we call it as androgenesis plant culture. Gynogenesis means similarly gynoecium part, female part like ovary, ovule, if I'm using to initiate the culture, we call it as gynogenesis. So haploid production can be done either by using andrecium part of a flower or gynoecium part of flower. Based on that, the technique can be labeled as androgenesis and gynogenesis. Okay, so this is how the androgenesis can be done directly. The anther in an unopened flower bud, you have to take it inside the leaf. You have to open the flower bud. Before that sterilization technique, as usually you have to do, take the anther aseptically, cut it properly, and you can plate it in the MS media. Proliferation happen and it will give you the plant. Or else, more precisely, if you want, means take the unopened flower bud, gently open it inside the leaf, cut the anther, and squeeze the pollen in the distilled water, give a wash and filter it in a paper. Then by using gentle sterile brush, you can take the pollen and you can start the inoculation and you will get the plant. And if you check the chromosome number, it is only half of the number because you are taking the germ cell before fertilization. Immature pollen you are taking. So these are the pollen division. There are 4 possible ways the pollen can divide and it will give you a complete plantlet. So this technique we call it as androgenesis. So moving on to the next technique. Similarly, gynogenesis I want to inform. You can remove the calyx, corolla, andrecium and gynoecium part alone. You can insert inside the MS media. Otherwise, make a transverse cut over the ovary and you can make the ovule to contact the media to proliferate the plant when you are using gynoecium, ovary or ovule to raise the plantlet. That we call it as gynoecium. Moving on to the next technique, that is organogenesis. Organogenesis is nothing but direct formation of plant organ without callus or embryo from any of the plant explant. We call it as organogenesis. So that can be divided into 2 types, direct organogenesis and indirect organogenesis. So direct organogenesis means from your explant, directly it is giving the plant organ and full plant is arising. We call it as direct organogenesis. Indirect organogenesis means through the callus clump, if the plant is emerging from your explant, that we call it as indirect organogenesis. So for this differentiation, growth regulator addition like auxin and cytokinin in a proper proportion it needs, and most preferably MS media, B5 whites media and all, preferable for the organogenesis. Moving on to the next organ culture technique, that is somatic embryogenesis. First, what is meant by somatic embryo? Embryo-like cells emerging from your somatic explant, we call it as somatic embryo. These are the somatic embryo. The technique, we call it as somatic embryogenesis. So this embryo also, you can take it and you can start your culture. As it is taken from the somatic cells, we call it a somatic embryo. If I'm using anther, ovary, and ovule, if the embryo is arising, I have to say it is zygotic embryo. Here also, direct and indirect somatic embryogenesis is also there. The main application is it is very easy. From this embryo, you can arise a plant and you can hardening it. And somatic embryos can be used for the production of synthetic seed, that is sodium alginate gel entrapment method. We are using it to cover it because it is a naked embryo. There is no seed coat and all because it is somatic cell. So you can trap it and you can save it as a seed for the future use. So these are the types of somatic embryogenesis. As usual, from your explant, somatic embryo is directly arising to give you the plant, that is direct somatic embryogenesis, or else from the clump of callus, the somatic embryos are arising. We call it as indirect somatic embryogenesis. Both technique can be possible by kind of cell, pre-embryonic determined cell and induced embryonic determined cell. That will play a very important role in the either it has to go for direct or indirect embryogenesis. These are few pictures. This is direct somatic embryogenesis. From the leaf explant, somatic embryos are arising. This is indirect. From the clump of callus, you can able to see the somatic embryos are arising. And moving on to the next culture, that is protoplast culture. What do you mean by protoplast? A cell devoid of cell wall. We call it as protoplast. So we'll be having only the plasma membrane. It can be done in two methods. One is mechanical method. Another one is enzymatic method. There will be a question why we need to remove primary cell wall, secondary cell wall, why we need to disturb the tissue system. Which means when you are cutting any tissue, though the cell wall is damaged, if the plasma membrane is proper, that also can give you a new plantlet in plant tissue culture. This is one choice. Without cell wall also, you can able to regenerate into a new plant. Moving on to the mechanical method. Mechanical method is very tedious because you have to put the cell in the hypotonic solution. Then hypotonic solution, what happened? Water inside the cell will come out. That means exosmosis takes place. It will be shrinked inside the cell wall. So by easily observing through the microscope, you have to cut and remove the primary cell wall, secondary cell wall. Then you have to remove the protoplast. It is highly tedious. Because of this, it is not in current use. We are moving on to the enzymatic method. So plant cell wall is made up of cellulose, hemicellulose, and pectin, pectinase. So when you are using enzyme, we can use cellulase to digest the primary cell wall, hemicellulase to digest the secondary cell wall, and finally pectinase to disturb the tissues. If you are using enzymes in a different steps, we call it as two-step method. All together in one beaker, if you are using for the digestion, that we call it as one-step method. So after centrifugation, the cell debris and the broken protoplast will be form a pellet. The viable debris will be in the aqueous solution. Viable protoplast will form the sediment. From that, you can take and you can check the viability of the protoplast before go for regeneration into a new plant. And how do you check the viability? First one is fluorescent diacetate staining method. So this dye, if you apply in your isolated protoplast, it will stain only the viable protoplast. Dead protoplast will stain red. This is the first picture. Second one, phenosaffarine stain is also there. It only stains the dead cells in red. So red one you can discard, which is not stained. You can take it as a viable one. Then exclusion of Evans blue dye, only it will touches the intact membrane so that you can screen it. And the measurement of cell wall formation. Protoplast is devoid of cell wall. Immediately when you take it, when you're inoculating, the first work of the protoplast will be regenerating its boundary wall, cell wall. So that also by calcofluor white, you can able to calculate. Then oxygen uptake of the protoplast. The cell is alive. It is going for metabolic activity, means it needs oxygen. So oxygen uptake by oxygen electrode also, you can check it. Then photosynthetic activity, you can check. So after this, the ability of the protoplast to undergo continuous mitotic division, that also you can observe under the microscope. So hope you students can understand what is meant by organ culture, what is organogenesis, direct organogenesis, indirect organogenesis, somatic embryogenesis, direct and indirect. Then what do you mean by protoplast, how to isolate it, and the techniques involved in the organ culture. See you all in next video. Thank you.